for today's session. Okay, so I'd like some signals to know if I'm being heard or not. Someone can unmute and say we can hear you, or just put a reaction so I can know if everybody can hear me. Am I being heard?
Yeah, I can hear you.
All right, thank you very much. So welcome to today's session. This is the first session in April, and it's interesting that it's also April Fool's Day, but this is not a prank, it's a real session. So tonight we're hosting a guest. His name is, please permit me, I hope I get the pronunciation of your surname correctly, sir. His name is Joel Omage. I hope I'm on track.
Yeah, you're on track.
All right, thank you very much. His name is Joel Omage, and coincidentally, he shares the same alumni with Martin and Wealth. He got his BSc in biochemistry, like many people in the community, from UniBen, and he has an MSc in chemistry from Western Kentucky University. So right now, he's a research scientist that is working with Novartis, and he has a lot of expertise in different areas like CRISPR, ELISA, qPCR. So today, he's going to be talking to us on GDF-15 receptor agonist as a potential treatment for obesity. And we'd like to very much welcome you, Sergio. Welcome to today's session. So this is where I will hand over and allow you to start your presentation. You can go ahead now.
All right, thank you for the introduction. Thanks for those kind words. I hope everyone can hear me clearly.
Yes, we can.
All right, so I'm just going to share my screen. Let's see. Can you see my screen?
Yes, we can.
Let me try to go back. All right. So I'm just going to put it in a PowerPoint mode. Okay, good. Just a second, trying to get the laser pointer. All right. Okay, all right, everyone. Thanks for having me. I'm going to be talking on GDF-15 receptor agonist, a potential treatment for obesity. So this is just a brief disclaimer. So every information on this slide is just primarily from my literature search and understanding. It's not by any means associated with my company or any ongoing research being conducted at the company. So the purpose for this session is to, of course, share about the drug development process, which is similar in all pharmaceutical companies. So I've decided to go with a drug candidate, which has a good potential, and of course my disease area of interest, which is obesity. So this is the overview of what I'm going to be talking today. So first, I'm just going to start with the drug discovery process, from drug discovery down to drug development. And then, of course, I'm going to give background information on obesity, the causes, the consequences, and of course, the World Health Organization intervention strategy. And then, of course, I'll go further to talk about the pharmacotherapeutic, option, which most pharmaceutical industries are, still conducting research on to develop more effective drugs for. I'll give an introduction to the target we'll be discussing today. And then after that, we'll talk about the preclinical study of the targets that has been conducted so far. We'll discuss about the clinical study, and then we will conclude. So, so this is an overview of the drug discovery and development process. of course, the first thing is to identify a target. So primarily, this is where I come in, where I currently work. So what we do is we're involved in the exploratory phase where we identify, a target, and then we perform in vitro studies to see the effects or downstream effect of the target. And then after validating that target, the next thing is primarily heat generation. So what that means, you have to like conduct some screening, after developing assay. Then you want to see if the compound, have any effect on the target, and if you are able to identify some heat, which have some, binding affinity into your targets, the next thing is to optimize the heat, and then you want it to have a very specific affinity for the target, and that is what is known as lead optimization. And so for the lead optimization, generally the point is to improve the absorption, distribution, metabolic, and excretion properties of the drug, and that is just primarily pharmacokinetic, and pharmacodynamic based experiments, just to make sure that the drug or the lead compound is safe for the organism. So you want to ensure that everything is not toxic before you begin to think about the development phase. And of course, the development phase involves two major steps, which are preclinical and of course clinical strategy. So I think for the preclinical, what we typically do in the industry is to begin to inject it. Of course, this it could either be compound, it could be peptides, it could be antibody, which you have identified to treat the disease you are interested in. And so you inject it into animal models, and then you also conduct pharmacokinetic study to ensure that this compound or this whatever the the lead candidate is safe, and has some efficacy. And then when you've gotten data which is safe and has some efficacy, you can then move into the clinical phase, for food development. So for the clinical trial phase, there are three steps. The first phase is you just work with about 20 to 100 candidates. So these are healthy candidates. You want to evaluate primarily the safety, of your drug, or your lead candidate you're working on. So, so if it is safe and everything looks okay, there's no, it doesn't have any toxicological, adverse effects, then you can move into the next phase, which is phase 2. So primarily for phase 2, you want to ascertain the efficacy. You do a dose range to ascertain the efficacy of your potential candidate. And if that comes out positive or successful, then you move into the next phase, which is phase 3. So generally you're going to be working with patients who have the disease actually, and then you increase the number from about 300 to about 3,000, and so if that looks good, then you can, then market your compound, after, of course, that would have, you would have secured approval from FDA, or the European medical agency. And after that, of course, there's also going to be like post-approval steps involved, which is just pharmacovigilance to ensure that this compound has, consistent, safety and all of that. So, so that is just the life cycle of the drug discovery and development process. So basically, I didn't talk about this quickly. So this is what most industries use, which is reverse pharmacology, to identify your targets. So this is like target-dependent, drug development. Previously, what they do was just forward pharmacology, which means you screen a number of compounds, and then when you see like a phenotypic, response, then before you start making attempts, to identify which target your compound binds to. But that is just basically a trial and error, process or strategy. And so these days, companies are more geared towards reverse pharmacology, which means you just have like an hypothesis, what target, is responsible or can be modified, to give you, the phenotypic, the phenotype you are looking for. And then you begin to identify, assays, develop assays, and begin to check, screen for compound. Basically that is, the strategy that has been used in top pharmaceutical companies. And of course, like I already mentioned, once the compound or the peptide or antibody have passed through all this process, it is then brought to the market, of course, pending approval. So let's quickly talk briefly about obesity, which is the our disease of interest. Like many of you already know. So for adults, obesity is defined as greater than or equal to 30 kilogram per meter square. So anyone who has a body mass index greater or equal to 30 is termed to be obese. So for children, it's a little bit different, but it's quite similar. So for children, it's about 33, that is 5 years and below. And then for kids that are between the ages of 5 to 19, it's about 32. So the standard deviation is about plus or minus 3 and 2. And so you can see the worldwide prevalence of obesity, nearly tripled between 1975 and 2016. So this is a major endemic. And of course, 39% of adults aged 18 years and over, were overweight in 2016. And of course, 13% of those, that statistic is obese. 39 million children under the age of 5, were obese in 2020. And of course, I saw in another paper that the projected number is going to go to about 300 million by the year 2025. And so you see that obesity is a major endemic, even in Africa today. When I was going through the World Health Organization website, it's like a double issue, for people living in Africa, due to poverty, due to malnutrition, and that could lead to, people just eating a particular set of diets, eating energy-based diets, and that will just increase, the level of fat and glucose in your bloodstream, which can lead to, obesity. And so this is a major issue. So what is the primary cause of obesity? The fundamental cause of obesity is just energy imbalance between calories consumed and calories expended. And so these are the two major reasons there is an energy imbalance. One of them is an increased intake of energy-dense food that is high in fat and sugars, just like I mentioned about, maybe children in Africa due to malnutrition, just eating mainly, food that are high in fat and sugars. And then another reason is decrease in physical activity. So these are the two major reasons. There is an energy imbalance rather between calories consumed and calories expended. So what are the health consequences? One of them is cardiovascular diseases. And of course, obesity, the department I work in at Novartis is cardiovascular disease. We've seen that most obesity patients, they are predisposed to cardiovascular diseases, and of course, these are some of the areas we actually focus in: heart disease, and of course, diabetes too, type 2 diabetes specifically. There's also, the tendency to develop musculoskeletal disorders, so this is also a big issue. And of course, even cancers too, especially prostate cancer has also been associated with obesity. And for children, who have obesity, the tendency for more severe, cases of obesity, is high, which could also even lead to premature death and disability in adulthood. What can we do to actually reduce obesity? Of course, there are three major things we can do, and this is part of the global strategy of World Health Organization. Just as simple as it's written right here. Basically, you need to limit your energy intake, from total fat and sugars, increase consumption of minerals, vegetables, cereals, and of course, engage in more physical activity. At the government level, they need to encourage policies, that will help improve healthy diets. For example, tax policy or sweetened beverages, can definitely go a long way to allow, the people to have access to healthy diet. And of course, food industry, they need to produce more foods that are low in fat and sugar and even salt content, which are major risk factors to obesity. And of course, they need to ensure that healthy and nutritious choices are valuable and affordable to all consumers. So like I said, this is the strategy of the World Health Organization. So generally people who are involved in weight management program, increase physical activity, control their diet. It has actually helped, in reducing their weight. But the thing with this program is it has a very low rate long-term success. And that is where the need for pharmacotherapeutics comes in. So there are some people who use the weight management program and of course incorporate, drugs, which offers a long-term treatment option, for weight loss, especially in chronic cases. There's a gold standard actually, because there are some drugs in the market, for the treatment of obesity. One of them is semaglutide, but the issue is, we've noticed about 5 to 10% loss in. It's good, but it's just like 5 to 10% weight reduction. And so, of course, there's still need for more research to come up with maybe better drug candidates that can help, to for the treatment of obesity. And that is where the focus of our study comes in today, which is on GDF-15. And so GDF-15 is a very popular target, because I think sometime in the early 2000s, it was found that, obese patients, have high levels of GDF-15. And of course, there was a study of a monozygotic twins. Of course, these are healthy, people. And then they also had high level of GDF-15, and they found an evens relationship between GDF-15 and body mass index. So a high level of GDF-15, correlated with low level of body mass index and vice versa. So what is this GDF-15? It's a growth differentiation factor 15 protein, which is secreted by various tissues, especially in the liver and even in the kidney as well. And of course, the heart. It belongs to the transforming growth factor beta superfamily of proteins. And of course, like I already mentioned, it has been associated with body weight reduction. And of course, even for the treatment of type 2 diabetes, some studies have shown that. And a high level of GDF-15 has also been associated with low level of food intake, because there's been GDF-15 relationship with GFRA, resulting in downstream signaling, which helps in increasing the satiety rate in individuals, which of course results in reduction of food intake. I'm going to show that in a couple of slides away. And of course, studies have shown that GDF-15 can modulate energy balance by reducing food intake and increasing energy expenditure. So the more you consume food, the more your body kind of like produce energy from that, which can help to reduce body weight as well. And the receptor for GDF-15 is GFRA. So this receptor is mainly found in the brainstem. So briefly, let's quickly look at how GDF-15 is transcribed and secreted in the body. So there are some external stressors that actually trigger the production of GDF-15. So one of them is amino acid deprivation. And of course, low oxygen levels like hypoxia, and of course the drug metformin, which has been used for the treatment of diabetes, can actually initiate the whole process. And this actually activates internal stressors, such as this kinase protein, PKROHROI, which triggers this activation factor ATF4. And this actually binds to target DNA, leading to the transcription of GDF-15. So GDF-15, what happens next is actually secreted into the endoplasmic reticulum. And of course, that is exported to the extracellular surface of the membrane. In the next slide, we'll see the signal process involved between GFRA and GDF-15. So what happens is upon binding to GFRA, GFRA actually recruits RET, recruits RET, and of course, this combination, GFRA, GDF, and RET actually result in decrease in appetite, which actually result in, reduction in weight, reduction in weight, and this is the whole idea about the signal process. So this is the target we are looking at. This agonist LY3463251. It's actually a potent and long anti-agonist for the GDF-15 receptor. It acts similarly like GDF-15. So what they did here was actually to synthesize a fission protein, which contains a monomeric IgG4Fc. So it contains two of them linked, to GDF-15. So basically, the IgG protein is a dimeric protein, but it's highly insoluble in the serum. So the idea was they actually synthesized a monomeric protein, which actually has a longer active lifespan in the blood. And so the idea was combining the GDF-15 to the fission to the monomeric protein actually increase the half-life of these drug candidates. And so that was the idea behind the synthesis of this agonist. The previous study has shown that GDF-15 has a low half-life in the blood, and that could affect pharmacokinetic response in obese patients. So after the synthesis of the agonist, so they wanted to actually show that this agonist also retained its binding affinity to its receptor, which is GFRA. And so basically, this is just a study to show that the rate at which GDF-15 binds to its membrane, and of course the dissociation, the rate of association was higher compared to its dissociation, as you can see. So it quickly binds to the membrane, and the rate of dissociation was also slow, the rate at which it dissociates from its membrane. So this study was conducted in human. I mean, this was the GFRA that was used was human GFRA, monkey GFRA, and of course mouse GFRA. But for the mouse GFRA, we saw that there was like a low affinity, for GFRA to the agonist. However, in the next study, we shall see the response of the downstream response between the GFRA to its ligand, which is GDF-15 agonist. So on the top right here, you find cells which was transfected with other families of GFRA. So this is not specific, directly specific to the GDF-15 agonist. So that is why in this graph, for the agonist, we do not see any response. On the y-axis, we see PEC, which is like a downstream signaling factor in the GDF-15 pathway. So PEC is actually activated, and we see a high activation of PEC in the cell when this pathway is activated. So we have vehicle, which of course has no, it's not transfected with any receptor. And this is a GFRA alpha-1 receptor. This is the agonist we're actually interested in. So upon adding this ligand or the agonist, there was no response. And that was because this cell is not GFRA. And of course, GFRA alpha-2 is also different from GFRA, which we're not interested in. And the same applies with GFRA alpha-3. So this is also different from what we are interested in. But if you look at the diagrams below, these are cells that were transfected with GFRA. And of course, upon adding GDF-15, you can see that there was an increase in PEC, readout, showing that this is highly specific. GDF-15 is highly specific, for GFRA. And of course, this native GDF-15, and of course, this other one signal or curve is for the agonist, which is similar to GDF-15, indicating that both candidates actually stimulate the increase of PEC. And we also see that too for mouse. So this is for human, and this also for mouse. So the next thing, that was just an in vitro study. The next thing was to carry out an in vivo study to see the effect of this potential agonist, GDF-15, and its association with body weight in mouse, in rat, and of course in cynos, which are monkeys. And so on this graph, this is just, I didn't show the data for the lean rat. So this is just diet-induced obese mouse. And of course, the data below is for the cynos, which are monkeys. So the C graph just basically shows, of course, the curve in black is just the vehicle. There was no, it was just subcutaneously injected with PBS, which is the vehicle or the negative control. And then of course we have different concentrations of our agonists. And then you can find out that within 2 weeks, as you inject the mouse, the mouse with our agonists, we notice a reduction in body weight. So this was like a drastic reduction in body weight. And then of course, food intake, I think this was about 26% from the study. And then for the daily food intake, it also shows that there was also a reduction in daily food intake, implying that the reduction in body weight was as a result of reduction in food intake by the mouse. For the monkey, we also noticed the same trend. So the vehicle is in black, and then the agonist, they injected subcutaneously into the sinus, 5 milligram per kilogram. And then there was also a drop in body weight, which correspond from a drop in food intake by the monkeys. So this study was conducted in 6 weeks for the monkeys, and then for the mouse, it was conducted in about 2 weeks. For the mouse, it was dosed every 3 days, and then for the monkeys, it was dosed once every week, 5 milligram per kilogram. And then before I go there, so what happened was after they obtained this data, they decided to perform a 1 month and 3 month study just to ensure that the drug has a good pharmacokinetic absorption, distribution, and excretion rate, and it wasn't toxic. And then they found that the optimal dose for the mouse in the 1 month study was about 0.03 milligram per kilogram. And then for the 3 month study, it was about 0.1 milligram per kilogram. For the mouse, so the dose range was about 0 to 180 milligram per kilogram. They didn't notice any adverse effects, for the monkey, even at 180 milligram per kilogram. And so that was also the optimal dose. For the mouse, there was some, toxic effects noticed, and so they had to stop at 0.03 for the 1 month study and 0.1 milligram per kilogram for the 3 month study. So having obtained that data, typically before you conduct a clinical phase 1 study, you want to ensure you reduce or you start with concentration that is like almost 1,000 fold, almost 1,000 fold dilution of your original optimal concentration. And so in this study, what they did was the minimum concentration was about 5-fold, no, was about 700 fold dilution from the obtained PK study or data. And then the maximum concentration was about 5-fold increase. So basically, they randomized about 64 healthy patients, and within 20 days, they randomized about 64 patients, and 48 were dosed with the agonist, and about 16 were dosed with the placebo, which is just, it could be PBS or something else. And then, of course, follow-up study, at the completion of the clinical phase, was about 42 days. So they conducted 2 parts, which is like single ascending dose and multiple ascending dose. And so what this typically means is for single ascending dose, you just increase the dose, with different cohorts, and then you observe, the safety profile, of the drug in this cohort, for the time frame, which is about, it was about 7 days for each cohort, 7 days, and I think they had about 8 cohorts, and with the maximum concentration being 24 milligram. For the multiple ascending dose, the major difference between this and part A, the single ascending dose, is that as you increase the dose, at some point you begin to up titrate. As you can see here, first you can just dose the heady candidate with about 3 milligram, and then every other week you dose with 6, and then with 9. And then in the next cohort, you dose with 3, every other week, 9, 15, and 24. So the idea is just actually to mitigate against GI, gastrointestinal intolerance. And so that is what they try to do with this multiple ascending dose design. So the agonist plasma concentration profile showed that about the average lifespan of the drug was about 72 hours, between 48 to 72 hours for the single ascending dose. And the same thing was also observed, or similarly, the lifespan of the drug was about, I think it was about 48 to 72 hours for multiple ascending dose, and then just 72 hours for the single ascending dose study. And so which shows that the drug has like a good lifespan, spending about 72 hours after dosing, in the serum of the individuals, which is enough time, to initiate any pharmacokinetic response. And so these are the results from the clinical study. So for A, it's just a time course of body weight change from baseline. And so this is the baseline, the placebo. Remember, for the mouse and monkey study, we saw a significant difference, between the baseline and, of course, the curve showing the dose response. But here we don't really see like a significant difference. This is just 0 to -1 between the baseline and, of course, the different concentrations of our agonist. So which implies there was no significant drop in body weight. However, if you look at the food intake, in calories, there was a good drop, significant drop in food intake, but we do not see it reflect in the body weight. So there was no like significant drop in body weight. And then, of course, we wanted to look at the visual analog score, which indicates like if the GDF-15 actually indicate or tells the brain, to reduce food intake. And so, of course, we see a high score, with the highest dose, which is about, I think, with 3, 6, and 9 milligram up titration. We see there was like an increase, in satiety score. And so this means that, of course, even though there was like an increase in satiety score telling the body to reduce food intake, we did not see any significant change in body weight. And of course, they couldn't really show the reason why this was the case. It could be maybe the native GDF-15, initiates, pharmacologic response different from the modified, agonist. So there's possibility that more studies also needs to be done, in such a way they can design the agonist to better like initiate, or interact with the receptor to bring about, a reduction in body weight. And of course, the conclusion is that, of course, the agonist shows that this GDF-15 that was modified could actually bring about reduction in body weight in rodents and cynos. In humans, however, we do not really see a significant reduction in body weight. However, we saw a reduction in food intake. So I think more study needs to be done, and I'm not quite sure why that was the case. But GDF-15 is really promising, and I think more study has to be done because, like from what I showed, they combined like 2 monomeric FC proteins to the native GDF-15 that could have played a role, and maybe other things could have happened. But this looks really promising. And I think more study has to be done. So thank you. If you have any questions, I'm ready to answer. Yeah.
All right. Thank you so much for that presentation. I think seeing the conclusion, realizing that it can reduce food intake. I think in the Nigerian system right now, I think that can reduce food intake most likely will have some form of effect in making people look a lot less obese. All right, thank you so much for your presentation. The floor is open for questions or comments. If you cannot unmute, please feel free to use the chat box. I'll read out your questions. And if you can unmute, you can please send a reaction so that I can tell that you want to. So if you check the reaction icon, you can see the option to raise your hand. So you can raise your hand if you have a question or comment, or if you'd rather use the chat box, the floor is open for that. So who's going first?
You can ask question ranging from, whether this research or anything about drug discovery development, any questions.
All right.
Or it's just like this.
Yeah, the Martin is saying thank you very much. Great presentation. So let's see if anyone has questions yet. So I wanted to find out, because obviously there's going to have to be more research on this. You
Absolutely.
mentioned that as well.
Mhm.
So do you personally have any plans of going deeper personally?
So you mean with my own research?
Yes.
Okay, so first of all, this is not my research,
Okay.
but I'm working on a target, which because of our policy, I won't be able to disclose. So I'm
All
currently
right.
working on that, and that is a different target, with the same
Okay.
idea to actually, for the treatment of obesity. So different companies, if we have so many targets we are working on, and the target I'm working on is really promising, but it's not going to be GDF-15 based. It's a
Okay.
different target, and we're also going to be designing. We're still in the early drug discovery phase. We've not even got into preclinical study yet, but we've had like in vivo data, in vitro data to support that target. Is going to really be fruitful in terms of developing a compound, a ligand that can activate it, for the treatment of obesity. Yeah, so we're currently working on a lot of targets, but I think another team is working on GDF-15, but that is not just within my jurisdiction. Yeah, so we'll see what, even from the study, they mentioned that, these studies just with this particular agonist.
Yeah.
Yeah, so of course, maybe depending on how it is designed, remember in the drug discovery process, they have to like design the drug in such a way that, it can better interact with its receptor to
Yes.
initiate the right response, so I think, with the drug design, this can also be further modified. And then of course we'll see, if it's going to like give. And another thing that even affected this study was, it was conducted in 2020. I think COVID-19 also affected the study because you are working with, people, many of them wouldn't want to even show up. So at some point, so
Yeah.
many candidates, at some point they dropped. And so that in a way also affected, the outcome of the study. Yeah, so I believe more studies will be done, and with the promising potential of these candidates, and I think with a little bit more modification here and there, I think it may just definitely translate, from the study we saw in the mouse and monkey into patients, human patient.
Yeah. All right. Thank you very much for that. On a lighter note, if the potentials are fully explored, it might put gyms out of business.
Oh,
So
really? No,
there are a lot of
no, you
people.
can combine both. It's even better. That will even be safer, because some of these drugs, they actually have adverse effects. I think that is why they do intensive study. There's so much money that goes into it, just to develop a drug, and this thing takes about 15 years, before you can ever come up with one. I think combination of weight management intervention, like going to the gym, which is rigorous physical activity, controlling the diets that you eat so that you don't consume too much energy. Food that is high on fat and sugar. I think combining both, programs would definitely help. Actually, because this is not just for people who are. If you are able to go to the gym, it means that you are not too obese. You are still good, right? There are some people who are. There are some people who are so obese that they cannot even stand up. So
That's
for those kind of people, they definitely need something form of a therapeutic drug, that will help them get in shape. And then, when they start, like, they are not able to get up and do something, and then they can even combine every other stuff, like going to the gym plus taking their drugs until they get to a point where they no longer need the drug. Yeah. So I don't think it's gonna. Yeah, I know. I know you were just saying that, but again, I think it's not gonna put out the gym business. Yeah.
but I think we'll know at the time when this gets to that peak. So actually we have questions now. So there's a hand that is raised. Um, Somebody's going to unmute and ask her question, after which I'll read the questions in the chat box. So Ruth, you can please unmute and ask your question.
Let me stop
Okay.
sharing.
Hi, Joel. Thank you so much for that presentation. I'm sorry I sound just like the person that you're talking to. We're twins, actually, just so you know
Oh,
that
okay.
yeah.
No wonder I was confused. I was seeing 230. I was like, I hope I pronounced the name well, by the way. I was
Uh-huh.
like,
Yeah. Yeah, great.
okay.
Okay, so I wanted to ask, and this is obviously because you are apparently in another country. And here in Africa, there have been conversations on weight stuff and everything. There's this thought that we are dealing more with poverty and people being malnourished and underweight than people being overweight. So being overweight sometimes here in Africa feels like a personal problem than an actual like issue that like a public health issue. I don't know how to explain it, but I think you could get the picture. So most of the time they tend to prioritize people that are underweight than people that are like overweight because they think they can just go and solve their problem. So with this research coming up, what I wanted to know was just your thoughts on how to create awareness in Africa around this. Because if somebody wanted to bring it to Africa, we'd first have to get people to be interested in it based on the kind of climate we are. So what are your thoughts on how to like create some form of awareness to support research like this for people that are interested in it in Africa?
Wow, that is a very, very good question. So you mean the research part, right? To create awareness for people to go into
Yes,
research
yes.
or okay. Okay. So first of all, I think the major issue is that the kind of research we do in Africa is geared towards phytochemical analysis. Correct me if I'm wrong. We don't really have, not many institutions have, state of the art, technology to carry out this kind of research. For example, I've seen right now I'm in the lab as I speak with you, on the 5th floor, you will see a very huge room. In fact, we have so many of them like that, where they have like high throughput screening machines, for, running assays to screen for compound. Whenever they are screening for compound, just for this GDF-15, just to eventually arrive at, your lead compound, you would have screened thousands of compounds. And these are things you cannot just do by your mere hands. How can you do that to screen thousands by using hands? It's not possible. So you need machine. So first of all, I think the government needs to be actively involved to create this awareness, and they need to pump money. For example, I skipped that part. About over $2 billion is invested just to, come up with a drug, just one drug. And this thing takes like 15 years, even in developed countries, right? And so what that implies is that first of all, the awareness needs to start from, this thing, we need to acknowledge that this is a major problem. Initially I know it's higher here, obesity, but again, I realized that even in Africa, due to, like you mentioned, malnutrition issue, people tend to also have obesity, right? So I think we need to acknowledge that this is a big problem. Government need to, act like a major stake, play a major role in this too by pumping, money into research institutions, into academic, so that students too will be interested, because many people are, even there's a lot of brain drain. A lot of people are leaving, coming here to do such study. We don't even have pharmaceutical institutions or industries in Nigeria that conduct research. What we have is just in marketing harm. For example, Novartis have a company, a branch in Nigeria, but they are not doing research. So their research center is just, secluded to, like, North America and a few places in Europe. You won't even find many other like pharmaceutical industries are into research in Africa. So that is a major issue. So I think government need to play a major role, and maybe non-profit organizations too, also need to see if they can, sensitize, people. But again, sensitization is not just enough. Like I said, this thing involves money, a lot of capital, right? So that is what I truly think. Government need to be active. We need to spend more money on education. What we are experiencing in Nigeria, a lot of strike, because there's no money to even pay, lecturers. Then where do you even find money to even invest in this technology? So that is a major issue. So I believe when government shows interest, of course, many people too will want to go into research in Nigeria. And you see, people will start addressing what are the real issues here, which disease area is a major issue. that we need to address. Obesity is one of them. I know malaria is a major epidemic too. So you see people wanting to do this research. I didn't come here wanting to do obesity research, but of course I joined Novartis and discovered that, oh, this is a big issue, and it's a big issue because cardiovascular disease is the number one, leading cause of death in the world. And obesity is a major risk factor for cardiovascular disease. And so what happens? You see most pharmaceutical industries, wanting to go into it. We need to identify target and stuff like that. I don't know if I'll be able to answer, but these are my thoughts anyway.
Yeah, that was really good. Thank you very much. Thank you. All right. Thank you so much, Ruth. Thank you, Joel, for that. It's very important. So let me just go to the questions in the chat box. Wealth wants to know, he's putting Q1, so I feel like maybe he has other questions. I don't know yet, but the question is, in your own opinion, what kind of modifications should be made or might be made to the agonist to improve its activity or the outcome on obesity? I don't know if you get the question, like in your own opinion, personally.
Yeah, I understand the question. So, so in my own opinion, so first of all, I think the GDF-15 itself has been used. So the first, I just want to give you a background. And then what they wanted to do, they noticed that the half-life in the serum, was very low. In fact, the concentration too, even when it's like highly expressed, it's about 0.1 nanogram per mil in the serum. And so the idea was, okay, what can we do to increase the half-life? Because increased half-life of a drug will also help, like increased pharmacology exposure can also increase, the activity of the drug to actually do its work, in reducing, maybe body weight and stuff like that. And so that was why they needed to add IgG-Fc. Because IgG-Fc, the Fc binds to a receptor, which helps to extend its half-life, which was a great idea, but again, one thing I want you to look at is there are so many other targets, that people are looking at. So not necessarily, oh, this is a very great target. I don't know if maybe they could, maybe they could remove, one of the, I'm not quite sure about what they can do, but in my own opinion, I would want to look at other targets. I want to look at other targets, that I've also shown, I've also been associated with, a decrease in body weight, upon, agonizing this, agonizing the receptor and stuff like that. Like the current target I'm working about, which I'm not going to, share due to policies. But for this particular target, I'm not sure what else can be done, because I think, increasing the half-life was already a great plus. They did fantastic, job there. And if that still didn't work, of course, we saw like a reduction in weight in animal model. It just didn't translate into the human model, So I think what I would even suggest that they should perform this study again. Because there was some drawback, there were some limitations. Even for the clinical data, we saw that there was an increase in body weight for the baseline at the end of the 12-week study, which is a little bit abnormal. That is a limitation. There should be no increase. So what could have happened? I would even suggest they perform the study again, and actually, just to confirm if this study is reproducible, if they are going to get a different result, because it looked great in animal model, it just didn't translate into the human subject that were used. So for me, I would say they should try that target, perform the study again, and again for the modification, mostly just chemists do that, so I wouldn't know what else they could try, except maybe just try different targets, yeah, that is what I would say.
Thank you so much for that, Joel. So Hassan, Ibrahim Hassan has another question, or has a question as well. So this is his opinion. He said, I used to think that there's some sort of public discouragement on using drugs that are supposed to achieve weight loss, like appetite suppressants rather, they were once banned. So what has changed over time that has warranted this level of interest now in going back to getting medications that can reduce obesity and all of that, since they were once banned at one point?
Funny enough, I'm not even aware of the ban. But again, um, like I mentioned, just weight management intervention program has not really helped, you know, and so other like therapeutic approach needed to, of course, if we take drugs for other things like malaria, take drugs for high fever, take drugs for cancer patient, take drugs. I don't think development of drugs too for obesity is going to be a problem, right? In as much as you can, show proof, of course, before they approve the drug, they will ask you, there's what they call, I'm trying to remember. Um, you need to show like a data, I've forgotten the name, show a proof that, oh, this thing has satisfied, past safety profile, it's also efficacious, there's no adverse effect, then I don't think that is going to be a major issue. There's even a drug, I've forgotten the common name for that drug, but it's made of semaglutide. Even it's trending on TikTok. A lot of people celebrate. Elon Musk took it, it reduced about 30 pounds. Kim Kardashian is taking it. A lot of these celebrities are taking it just to keep fit. And so it's not causing any issue. And so if these drugs are working, and there's a lot of people, like I said, it's projected to be up to 300 million obese people in the world by 2025, and just gym alone, normal intervention, but it's not going to help help the situation. So there has to be like drugs. And even with this current drug, even with the semaglutide, it doesn't reduce as much. The weight loss is about 5 to 8%. So we even need more potent drugs. And that is why the interest has gone up, So if there was a ban before, which I'm not sure, but I think the problem, is just overwhelming. There's nothing people can do anymore. People are dying. The death case, cardiovascular is the leading cause of death. Something has to be done. If drug can help us intervene, why not? So, yeah.
All right.
Thanks.
Thank you so much for that, Joe. Actually, Ibrahim Hassan is not here, but actually, I think there was a certain appetite suppressant that was banned because of the fact that it had cardiovascular side effects. It was just one particular drug. I'm not sure it was generally, which I'm not aware of. But thank you so much for that. So Tolu Omojola has a few questions. I think I'll take the questions one after the other. So the first question he has is that, is it safe to say that we have a lot of predisposing risk for obesity in Africa than other parts of the world?
I don't think so. I think the statistics do not say, the statistics shown indicate that there's more obese people even in North America.
Yeah.
But it's just that it's increasing too in Africa because of poverty. You saying something?
No, you can go ahead.
Oh yeah, so there's increasing risks, because of also the poverty level. Malnutrition too is like a double-edged, issue, which means that people kid. I remember back then, when there's no food, you drink garri in the morning, in the afternoon you eat eba, in the night, it's just carbohydrates. What do you expect? So I think that is a major issue why it is increasing. But compared to the other part of the world, I don't think it's a major problem, but it's a big problem too, but compared to America, for example, it's not, we don't have more obese people in Africa compared to other part of the world.
All right. Thank you so much for that. So second question is that, is it possible that there are proteins that are competing for the binding sites in the study?
No, it's actually specific. So like I showed, okay, I've already put off my slide. So my very first data slide, I showed that it was specific, even for proteins from the same family. Protein from the same family, couldn't even bind, to the GDF-15 receptor, showing that the GDF-15 receptor was very specific, to the GDF-15. So there's no other proteins that are competing for the binding site.
All right, thank you so much. So the last question from Tolu is that, is there the chemistry of everything that you just talked about, is there anything in it that could be related to the maybe mechanism of action of slimming teas that are being sold in the markets now? Is there anything that can tie the two together?
To be honest with you, I don't know how the pathway, how limiting works. I may have to go read about that so I can be able to juxtapose if there's anything similar. So I know how this works, but I don't know how limiting works. So I wouldn't know if there's anything similar in their chemistry. But that is a good question. I can check.
Thank you so much. So Wealth has released question number 2. And the question is more about working in the industry that you're in. And he's asking if you could share some of your experiences. Is it more liberating? Is it more freeing to be in industry as compared to being in academia or academic research? Like research in industry, is it more free? Is it more liberating compared to academic research? And then could you share some of your experiences from working in industry as a researcher? Okay, I don't know. I think I've lost Joel. Can every other person hear me?
Yes.
So we can.
Okay, so I think we should just give him a few minutes. Maybe it's okay, I think he left the meeting. Okay, so let's just, maybe someone should reach out to him and let him know, or maybe trying to rejoin. So we'll wait for him. Okay, while we wait for our guest, I think we have a few announcements, so I'll just drop the announcements. But we're waiting for our guests because there are still questions pending. So I'm sure he'll join us shortly. So first of all, for the undergrad.
Can you hear me?
Oh, fantastic. Hello, Joel. Can you hear us? Can you hear me rather?
Yeah, I can hear you.
All right. So I was asking World's question actually. And I don't know if you remember what I was asking or if you
Yeah,
got the question.
I remember your question. Yeah, so you're asking if it's more liberating, to work in the
Yes.
industry as compared to academia. So I
Yes.
think first of all, it depends on your passion. There's some people who will tell you it's more liberating, that you have more freedom, you do stuff at your own pace. Because in the industry, we have like there's always going to be team. It's not that there's no team goals too in academia, but everything starts from the top down to the bottom. So, for example, the president or the leadership might just want to work on siRNA targeting. We want to work on RNAi. We want to work on just antibodies. So whatever they say, it will trickle down to the basic scientists. And so what that means is that we have a goal. And this year we have a goal. There's a team goal, there's an individual goal, and you have to meet your goals, right? And so it's kind of like it's a heavy pressure anyway. So it depends. For some people they like it. In academia, not like it's just you on your projects. So if, for example, it depends if you're a research assistant, you meet with your supervisor, it tells you this is what I'm working on, and you just between the both of you, and then you begin to do what you're doing. And so there's pressure, there's a heavy pressure on both of them. But again, it depends on what you really like. But for me, I chose the industry because I felt like, working in the industry is more translational. You can easily translate science into something substantial, like I'm working on drugs. So what I'm doing, I know that at the end of the day, it could potentially lead to the development of a drug that can be used to treat people who are suffering from any form of diseases, in this case, obesity, right? But in academia, you just work and whatever you find, you publish it. Scientific problem. That is all where it ends. And sometimes, even this, although you'll be like, oh, I've contributed to knowledge, but these things may just be there and nobody will even really translate, except maybe somebody see, oh, you did something interesting and they want to continue on that kind of research, right? But most times, even when we are working in the industry, we still read papers. We read papers like from Nature, and sometimes these studies are not even reproducible, and so they are just there, in the literature, but you cannot really make anything out of those publications. So, but here, what we do is judge towards, saving lives, helping people. And so that's why I think it depends on, what you're doing, if you are passionate about it. So for me, it's more liberating to work in the industry. I am enjoying the ride. I'm enjoying everything I'm doing. I feel like I'm helping people, by, participating in groundbreaking research and stuff like that. So I've worked in both worlds, and I can compare, and I enjoy it more here. And even in terms of course, you didn't ask me about this, for some people, there's more reward, both financially, working in the industry too. But for the liberating part for me, I would say it's more liberating in the industry. For other persons, it might be more liberating for them in academia. Did I answer your question, or was there anything more?
I think you did, but what else you can put in chat box but I think you answered pretty well. Thank you so much for that, Joel. And your presentation, everything, I think we've gained a lot. So I hope we would be able to get a copy of your slides.
Oh, okay.
For
that's
proper
fine.
digestion.
Okay, that is fine.
All right. Thank you so much for that. So I know everybody enjoyed the presentation, and hopefully at some point we'll still have you back. Maybe you tell us about the other targets that you're working on at the time. Maybe at that time it will be
published.
Yeah,
maybe they'll come up with the drug and so publish our data.
Sorry for a breach in etiquette, Rachel. Please, can I quickly say something? Yes, please. So I was going to say that it would be nice to have you come and talk to us about how you got into Novartis in the first place. Yeah, I think a lot of us would like to know that too.
I thought you guys were going to ask that question since. Is that going to be a separate session?
I think, well, sorry, Ruth. I think that it's because we would like it to be a separate session so that all the processes involved would be explained and maybe other examples properly given. So, because I feel like if you tell us now, you just tell us a story and we might not know the details I would want to know and carry our jotter and our books properly. I don't know if I'm speaking anybody's mind. So maybe we should look at scheduling another session with you where you tell us about it properly and then you give us all of your guidelines for free.
All right, no problem.
All right. Thank you so much for that. Thank you for bringing that up, Ruth. I know that some people want to be innovative for many other reasons. All right. Thank you so much, Joy, for your presentation. Thank you, everybody.
Okay. So yeah, this is a really great platform. I would like to say that. I like the spirit behind it, to learn. Even here at Novartis, I'm also part of a journal club where we discuss, recent, discoveries in science. We look at papers from Nature, endocrinology, and all of that. We just want to be up to date with the latest discoveries. And what you guys are doing is also similar. Getting abreast with the latest innovations. It just kind of help you stay informed. So I will encourage you guys to keep doing that. Connect with more people. There are so many of us, Africans, who are doing great stuff, in different parts of the world. Even some of them in Africa too. Try to stay connected, get them to come share their thoughts, and you guys can learn a lot, just like you guys are doing right now. And of course, stay connected with them too, on all social platforms. You never can tell. There might be an opportunity, and, people share opportunity with those who are within their cycle, those who are close to them. I can just share information with somebody who's part of this group, and then he or she will share it with the rest of the team. Oh, you guys should, try your show. There are people who haven't getting, jobs right from wherever they are in Africa, whether in Nigeria or that part of Africa. This is a result of network. So networking is key. Build your LinkedIn profile. Whatever you can get, more skills, on Coursera. Anyway, keep developing yourself, keep advancing if you're really interested in biochemistry. And if you're interested in working in industries like this, like I said, this is going to be helpful. And sessions like the one you guys are going to plan, next time will be really helpful too, So yeah, keep up what you're doing. Great job, so I would just encourage you guys to keep doing what you're doing. Yeah.
Thank you so much for that, Joe. I think that's very encouraging for us. And we're not saying goodbye, actually, because we already have a session booked down already. So we hope to see you again next time. We'll get across to you and then finalize the whole thing after deliberations. So on that note, thank you everyone for attending this session. Thank you for your time and thank you for the contributions and questions. Thank you again, Joel, for your presentation. So let's do well to enjoy the rest of our weekend and the rest of the month as well. And I know I'm going to enjoy it personally because Arsenal is going to win the Premier League. So, all right. Thank you, everybody. Good night.
Good night.
Night.
Good night. Thank you. Good night.
Night.
Good night, guys.